Okay, so this is the structure of a polynucleotide of DNA. We're zooming in. If we, go back up and look at this diagram for a DNA double helix, we're just zooming in on one strand. We're leaving out the other strand completely, and we're showing you what the atoms are like in that single strand of DNA. So there's a 5' end, and the first nucleotide, the first base or first nucleotide in a sequence always has a phosphoester on the 5' end, okay? So that functional group there is called a phosphoester, and that's sort of how you know where the end of the DNA begins. That's the 5' end. Then there's the sugar and a base, and then the 3' hydroxyl. The 3' hydroxyl of one ribose ring is linked to a phosphate that is also linked to the 5' hydroxyl of the next ring. This is an unusual functional group. We haven't really seen it before. It's a phosphoester on both sides, and it's called a phosphodiester. So, and you can see that that phosphoryl group in the middle has one negatively charged oxygen and then two OR groups. One of the OR groups is the 3' hydroxyl of the previous ribose ring, and the other OR group is the 5' hydroxyl of the next ribose ring. So the linkages between ribose rings in the DNA backbone are phosphodiesters. They're about as stable as phosphoesters. So, and this is important because your DNA needs to last a long time, right? Your body has ways of repairing it, but in terms of functional groups, it's about as stable as an ester or or just as a regular phosphoester. Okay. So as you go along, you can see here is the C nucleotide, then the next A nucleotide, its 3' hydroxyl is in a phosphodиester with the 5' hydroxyl of this T nucleotide, and its 3' hydroxyl is in a phosphodиester with the 5' hydroxyl of this G nucleotide, and then at the end, at the 3' end, you just have a free hydroxyl, okay? And that's what we mean when we write out a DNA sequence. We'll always write it from 3' to 5'. When you write a DNA sequence, you really have to indicate which side is the 3' end and which side is the 5' end. So if I were going to summarize this nucleotide in a linear sequence, this polynucleotide, I would write a 5' with a dash, and then I would write CATG. And I can write nucleotides left to right or right to left as long as I label which end's the 5' end and which end's the 3' end. If we were going to write a complementary polynucleotide, that hydrogen bound with this sequence, what would it be? We would start with the 5' end here. What goes opposite G? C. What goes opposite T? A, then T, and then G. So this would be the double-stranded, this would be the complementary sequence. Notice on the top, we're right to left to right, 5' to 3', and on the bottom, we're right to left, 5', 3'. That's how you write things out. Yeah, go ahead.
So at the beginning at the top, you showed it with like, we had 3 phosphates in the front of the chain.
Right.
And then at the adenine, is there always going to be 3 phosphates in that first phosphate chain?
Yeah, great question. At the beginning when I showed you nucleotide structure, I started with a triphosphate because you'd seen ATP before. You'll notice that in the sequence of DNA, that triphosphate's no longer there. It's just one phosphate. So we do need to talk about how DNA is synthesized. And we're going there next. Other questions before we do? So one skill that's going to be useful to you is if you can write out a sequence that's complementary to a sequence that you're given. And then notice, if I want to write, we'll call this one on the top the template, and then this is the complement. If I write out the complement in this direction from 5' to 3', that complement is CATG. So you might wonder, looking at this sequence, if I write out CATG like this, you might be like, oh, that's not complementary, because the complementary sequence is GTAC. Remember, you always have to write it 5' to 3', whether it's left or right. Do you see how this and this are the same thing? And you actually see how this sequence is self-complementary. Palindrome, yeah, self-complementary sequence. It's not the exact definition of a palindrome in the sense of like words that are the same forward or backwards, but it sort of is, right? Because it's self-complementary. Okay, I want to show you how DNA, the chemistry that DNA uses, the DNA polymerases use to make a nucleotide, a polynucleotide. So let's assume this situation is completely unrealistic, but let's just assume we've got a section of double-stranded DNA that we're zoomed in on, and the section that we're zoomed in on is CATG, and then the section that's being synthesized is CAT, and then here is the loose 3' end of the molecule, and we want to incorporate the last nucleotide, okay? So what I'm going to do now is we're going to zoom in on the blue strand that's being elongated. I'm going to draw the ribose ring, and this is DNA, so here is the 3' hydroxyl group. Here is the thymine base. Here is the 5' group that's involved in a phosphoester with the previous oligonucleotide, which I'm just going to do squiggly line and R. That's the rest of the molecule. Okay, so what I've really drawn here is just this T nucleotide, and this OH group, the 3' hydroxyl group, is going to be the nucleophile in our reaction. What's going to be the electrophile? Well, the next amino acid is what? I'm sorry, I'm saying it wrong. The next nucleotide needs to be what? G, right? So the thing that's going to allow that to happen is the molecule DGTP, which looks like this. Again, you've got the ribose ring, you've got the G base. The DNA polymerase will know to put G in this next spot because of the what I called above the template strand that has a C at this position. Then the DGTP, what does that look like? Well, here's the 5' hydroxyl group, and then this is just your typical deoxynucleotide triphosphate. And this one has all kinds of reactive phosphoanhydride bonds on it. We're used to this kind of thing. We've seen it before a ton, okay? And you know that nucleotide triphosphates have 3 phosphate groups, alpha, beta, and gamma. What nature's going to do is a basic group from the enzyme is going to deprotonate the 3' hydroxyl that's going to act as a nucleophile, and that's going to attack the alpha phosphoryl group. That'll be the phosphorus. This is the phosphorus oxygen, and all of this over here is called the pyrophosphate leaving group. It's a good leaving group in its own right, and then as you learned, it can be hydrolyzed spontaneously to 2 phosphates, and that creates, that makes it really energetically downhill. So the 3' hydroxyl, after it's deprotonated, attacks the alpha phosphorus of DGTP. That's going to generate an intermediate that looks like this. Where that OR group from the 3' hydroxyl is bonded to a phosphorus. That phosphorus should now be bonded to five different things. One of them is the 5' hydroxyl of the ribose base of G, and then the other is this pyrophosphate leaving group. So this is the pyrophosphate leaving group. And here's the phosphoryl group, and here's the kicker off for oxygen. That in the next step, electrons are going to kick down and kick off the pyrophosphate leaving group. And in the end, that's going to make the new phosphodiester bond between the previous T nucleotide and the next G nucleotide. So here is that previous T nucleotide. Here's the 3' hydroxyl that was just the nucleophile. Here's the phosphoryl group that got attacked and then kicked off pyrophosphate as a leaving group. And then here's the next, running out of space a little bit, here's the next ribose ring with a new 3' hydroxyl that's now ready to be a nucleophile in the next reaction. So that overall is how we're going to do DNA synthesis. The energy required to make the phosphodiester bond is going to be paid for by liberation of a really good leaving group, which is called pyrophosphate, which I'm going to abbreviate as PPI, and later that gets hydrolyzed to two phosphate groups, making the overall reaction quite downhill. And it takes nature a lot less time to do that than it did for us to talk through it. Essentially, RNA is going to be made in the same way. You need, in the case of DNA, your ingredients for DNA synthesis are DATP, DGTP, DCTP, and DTTP. Deoxytriphosphate versions of the various nucleotides. For RNA synthesis, the process is going to be exactly the same. Or very similar, only the ingredients are going to now have a 2' hydroxyl. So this will be ATP, GTP, CTP, and TTP. Your body makes all of these. It can use ATP to transfer phosphoryl groups to the various nucleotides to give you these nucleotide triphosphates. And those are the ingredients of DNA and RNA synthesis. Okay, questions about that? Yeah?
What's the reason on phosphate groups? What's the reason behind the carbonyl and the electron-rich oxygen being on the same side? Is it just to represent better?
What's the deal between these two being angled in the same way? Yeah, the bond angles here for the phosphate group should be roughly tetrahedral. I've drawn it this way so that I have plenty of space to draw the ribose rings and the bases. Yep. Okay, others? All right, let's see how good you are at writing complementary sequences. This is not from your quiz, this is from something else. I'm going to write a DNA sequence, and and I want you to write the complementary strand of that DNA sequence. So the template strand is here. I'm going to write it 5' to 3', and it's going to take me a while because I think slowly. TCA, ATC, CCA, ATT, 3 T's after that, then an ACC, then an ACT, then a TCG, then an ATT, then an AAG, and then a CAT, and that's the 5' end. Okay, so go ahead and write the complementary sequence. Just remember A's match with T's and G's match with C's. You have to think a little bit to do it. Nature doesn't have to think to do it. Nature uses hydrogen bonding and shape to put the right nucleotide triphosphate in the active site to be complementary, and then insert the right base and do the reaction. DNA polymerases are not 100% faithful. That means they sometimes put in the wrong base. Your body has DNA proofreading enzymes that come back in and recognize that something's wrong here and clip out the wrong one and put the right one in. And that's important for integrity of cell replication. Go ahead.
Did you say for DNA or RNA?
For DNA. Yep. All right, so that's pretty straightforward. Here's the answer if you've done it. Did I get anything wrong? So this is interesting. In DNA, there are 2 strands, as you know. One will be called the template strand, and the other is called the coding strand. The coding strand is the one that has the same sequence as the mRNA that will be transcribed from this particular gene. Only in DNA, all the T's, in DNA, all the things that would be U in RNA are actually T. So this matters because if you're going to read DNA sequence to try to determine what protein it encodes, you need to make sure you're reading the coding strand and not the template strand. How do you know which one the coding strand is? Well, you look for what are called start codons. So a codon is 3 I'm trying to figure out what's going on back there. Did I do something stupid?
No. We did something stupid.
Okay. I kind of wanted to be in on all the chuckles. I'm sorry.
No, I'm sorry.
So what you do is you look for a start codon. In reality, there's what's called a promoter sequence up here, further up on the 5' end of a gene that encourages a DNA or an RNA polymerase to bind there. But effectively, you look for a 3-base sequence that is the signal to for a gene to start. And the start codon across a lot of different life forms in RNA is the codon AUG. Or in the DNA coding strand, that would be ATG. So the presence of this ATG sequence here indicates that that is a coding strand for And what nature will do is it will use the template strand to prepare an mRNA sequence. So what I'm going to do now, and I'm not great at explaining this because I'm not really a biologist, but we'll do sort of poor man's animation, which is copy and pasting. We're going to show you a little bit of how RNA replication happens. There are two different kinds of RNA. One is mRNA and the other is tRNA. M stands for message. A messenger RNA is a transcript of the DNA. It's a copying of the DNA sequence that will then go to the ribosome and be converted into, serve as instructions for making protein. When a gene needs to be expressed, transcription factors, RNA polymerase, and enzymes come together and they locally unfold the DNA double helix, exposing both the template strand and the coding strand. And there's machinery that directs the RNA polymerase to start transcribing the sequence. And it actually starts way upstream of what we're going to call as the start codon. But let's just start here. It will start at the 3' end of the coding strand, and it's just going to start inserting mRNA, which we'll represent in red. Okay? So for mRNA, every time there's a T, we insert A. Every time there's an A, you insert U. Okay, so otherwise it's going to be exactly the same. U, U, A, U, C, G, A, G, U, G, G, U, and then a whole long string of A's, 2 U's, 3 G's, an A, U, U, G, and A. And this will be the 5' end of the mRNA and then the 3' end. So that's the mRNA message that gets transcribed. And then that goes to the ribosome for translation, and the RNA polymerase machinery leaves and allows the two strands to get back together. Notice that the sequence of the mRNA transcript is identical to the sequence of the coding strand, except all the T's are U's. Does that make sense? Yeah.
What is U?
U is uracil. If we go back to the bases, uracil is the version of T that's in RNA but not DNA. Uracil is just like T, lacks the methyl group. Yeah.
So I just want to double check because this has always confused me, but so if you want to test or give us this template strand and say like what is the mRNA transcript from 5' to 3', we'll want to first like write down the coding strand basically and then flip it backwards.
Yeah, so if you were wanting to transcribe this sequence, and there's an example of this on the last page of your quiz, but we'll go through this simpler example first. You start with the template strand, and in this case I would just give you the template strand. You would need to write the coding strand, remembering that this end is the 5' end. And then if you want to, you can divide the mRNA transcript into 3 letter codons. And you don't even have to rewrite it as long as you remember that RNA translation. So the process of making, sorry, a lot of terms we're introducing here. The process of making mRNA from DNA is called transcription. The word transcribe needs to take a message that already exists and copy it down. And then the process of going from mRNA to protein that's done in the ribosome, that's called translation. It's called translation because it's not a word-for-word transcription. You've got to take the information in the RNA and turn it into something different. So once you've got the RNA transcript, you want to look for a start codon, in this case red 5' to 3' AUG. Now we can do this right to left, but you may want to just write it down again left to right, because at least in English we're used to reading left to right. So what I will do is I'm going to rewrite now the mRNA transcript from 5' to 3' here. It's AUG, and as I'm doing it, I'm going to separate out the three letter segments. These are codons, and these are read 3 nucleotides at a time by the ribosome, and we'll explain more about how that's done a little later. But I'm seeing so far for our sequence AUG, CUU, AAU, CGA, AGU, GGU, triple A, AAU, UGG, UAG, and then UGA. And you can space them out 3 bases at a time, and then this is the 3' end. Okay, you with me on how we did that? We just reversed it. Okay. So how does that turn into a protein sequence? We'll learn about the mechanism of that probably next time. I'm not really ready, and we probably don't have enough time to talk about ribosomal protein synthesis. So we're gonna talk about that more next time. But translation is not hard as long as you have the cipher, or the translation book, or the Urim and Thummim. And in nature, this is our Urim and Thummim. This is the genetic code. This lists all possible codons. Now remember, each codon is composed of 3 bases, or 3 nucleotides. There are 4 different nucleotides. So 4 times 4 times 4 equals 64, 64 different codons, okay? And this is how you read the this is how you read a codon table. Over here on the left is the first base, the 5' side for a codon. Across the middle is the second base, and then in subdivided rows in each cell is the third base. So if I wanted to see what AUG was, I would find A, then I would navigate over to the right to find U, that's here, and then I would navigate downward to find G, AUG, and that tells me that's methionine, and it's also the start codon. The start codon is what tells the ribosome start making protein now. And that is methionine, and so if we want to translate this into a protein sequence, we start at the start codon, and right below I'm going to write the translation. And interestingly, proteins are synthesized from the N terminus to the C terminus. So this methionine is going to be the amino terminus of our new protein. So then we need to find what CUU is. So we go to C, navigate over to U. CUU, that's leucine. If you look at this codon table and study it for a while, you can see that we only have 20 amino acids, but we have 64 codons. So multiple codons encode the same amino acid. For example, if your first 2 letters are C and U, it doesn't matter what the third one is, it's always leucine. And there's some biophysical reasons for that we don't have time to get into, but there's really important reasons why there's flexibility at the third nucleotide. In part, this protects you 1 out of 3 times from the impact of a mutation. If at a particular point in your genome it's a U and it gets mutated to a C through random error, doesn't matter, your body's still going to put leucine at that position because all of the codons that start with CU, independent of what's at the third position, all code for leucine. So this builds in some protection against mutation. AAU, that would be A here, and then U, that's asparagine, that's N. CGA, let's see, C, then to G, then down to A, that's arginine. What I was just talking about with leucine applies to arginine as well, and this is called degeneracy of the genetic code. That doesn't mean the genetic code is like morally degenerate. Degenerate means equivalent mathematically. So the degeneracy of the genetic code means several codons code for the same amino acid. Again, this is protective evolutionarily against mutation, because if you change the third base in this codon to something else, it doesn't matter. You still get the same answer in translation. Does that make sense? Questions so far? Yeah.
We have this chart on the test.
Yeah, you'd have this chart on the test. I could make you memorize the genetic code. That would be evil because I don't have the genetic code memorized. And also, nobody does this by hand anymore. We all ask the computers to do it. But I'm preparing you in the event that our AI overlords deny us access to computers, so that if you've got it written down somewhere, you can still do science. Yeah?
So it starts with methionine, but then it ends with nothing. That's a stop codon.
Right, so the stop codon you will see is UGA. And if you look here, U, then G, then A. That is a stop codon. Why is that a stop codon? There is simply, you simply don't have a tRNA that recognizes that and gets loaded with an amino acid and goes to the ribosome. So the stop codon says stop simply because there isn't anything in the ribosome that can bind that and insert an amino acid there. Okay, so you do the next one. That's AGU. Which one is that? Serine, that's S. See how fun this is? See how much we're practicing at once? We're practicing DNA, RNA, and we're practicing proteins. In a minute, I'm going to ask you what the charge is on that thing at pH 7. GGU. GGU is glycine, good. AAA. So now we've got another joke, another peptide joke. The joke about what is the pirate's favorite amino acid is, it's arginine, of course. This one's a nested peptide joke. What is the automobile aficionado's favorite amino acid? It's lysine because it's encoded by AAA. Not that funny. I told you, right, that my oldest son, Nate, is taken to rating my dad jokes. Like, I'll say something, and he's funny because his are just as bad. He was trying to tell some jokes on Sunday. We were going to my mom's house, and he had to explain them to us. And I'm like, that's a good dad joke. I love that. Anyway, yeah, he'll say, that's a 3 out of 10, Dad. And I'm like, joke's on you, because that's what I'm going for. If you get it grown, that's actually what we're going for. Okay, AAU is asparagine. That's another N. UGG, UGG. What's that? Tryptophan. So maybe we can memorize UGG because when Elmer Fudd is frustrated because Bugs Bunny has escaped him again, he says UGG. And then tryptophan is his, never mind. UAG is, did we get that one right? Was that really UAG? Oh, I transcribed it. It's, that one should be GAU. I read it backwards wrong. Thank you. GAU. So that is what? Aspartic acid, D. Okay, then we get to stop. We're done. This sequence encodes a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 amino acid peptide. If I were Dr. Savage, I would have given you the DNA sequence and then told you to draw the peptide. So you'd have had to transcribe it to get the coding strand, to get the mRNA, and then translate the mRNA, and then you'd have to draw this thing. For our purposes today, since we spent plenty of time, actually on his exam, it would be draw it in a beta term. Like, isn't that evil, right? So for our purposes, let's just say what charge it is at pH 7. We got pluses for lysine and arginine. We got a plus on the N terminus. We got a minus for aspartate and a minus on the C terminus. Did I miss anything else? So at pH 7.4 or whatever it is biologically, I count 1 plus. Okay. All right. Quiz time. So I want you to get started on this. Transcribe the RNA. This is the coding strand. Write down the mRNA sequence from 5' to 3'. Start at the, so for your quiz, you'll start at the 3' end of the coding strand and go backwards. Your mRNA transcript should begin here. If you want to, you can rewrite it. If you're good at reading backwards, right to left, then you don't have to. But go ahead and write down that sequence. We won't have time to translate it, but I'll have you do that maybe just practicing as a practice between now. Let's be honest, you're not going to do it. We'll just do it because you got an exam today and tomorrow, right? We'll just translate it tomorrow, but get started on the transcription. For fun, convince yourself that the first 3 letters are a start codon, AUG, and the last 3 letters are a stop codon, UGA. How can you tell UGA is a stop codon? Because the ribosome, when it reaches that, is disappointed and surprised, so it goes, ugh, ah. I got nothing. Just use the codon table. I'm gassed. Ugh, I'm gassed. That's a good one. Yeah, I'm gonna remember that one. Thanks. Yeah, go ahead.
Can methionine show up in the middle of a protein?
Yes, methionine can definitely show up in the middle of a protein, and that's fine. It's just the first methionine in the sequence that signals begin.
Would it ever happen that the ribosome would latch onto the wrong methionine and start coding in the wrong place?
Yeah, is there a problem with the ribosome attaching to the wrong methionine? That is an excellent question, and I don't know the answer to that. I think because of that, methionine is one of the rarer amino acids, but yeah, I'll need to look more into that. There may be other things. What we haven't talked about is how an RNA transcript is recruited to the ribosome, and there may be other things other than just randomly scanning through the mRNA that says where it should begin. So we'll have to look at that. Okay, well, you got started on that. I'll post the quiz online so you can continue on it. We'll also work on it next time. Good luck on your exam today and tomorrow, and I'll see you next time.